Hello everyone. So thanks for inviting me for talk in this great conference. It's my first time to be here. Actually, I can see actually a lot of people have a very diverse background. So I'm myself is a molecular microbiologist. I'm working on the bacteria for the variety of things like protein transportation, outer membrane biogenesis, secretion stuff. So today. My name is Xiaolong Liu from Ben Berks lab in Oxford University. Today, I'm going to share you a story which has been published recently. It's about the biogenesis of the outer membrane protein in Bacteroidota. So, uh, as I said, we work with bacteria. Bacteria are a single cell prokaryotic organism that represents a very one of the most abundant life forms in the world. So they all, despite the differences they have, the phenotype, they all enclosed with a cell envelope, which separates the cellular content from the surrounding environment. So based on the structure differences of the cell envelope, bacteria are classified into two groups: the gram-positive and gram-negative bacteria. So for the gram-negative positive bacteria cell envelope, it contains a cytoplasmic membrane and a very thick layer of peptidoglycan cell wall. Well, in contrast, gram-negative bacteria, the envelope contains not only these two components, but also a unique outer membrane components. So the outer membrane is the first line of defense for gram-negative bacteria. It's very important and essential for their growth. That's why there are a lot of researches carry on the outer membrane, and it's like also central of the microbiology studies. So in the outer membrane of gram-negative bacteria, there are a lot of proteins present. So based on the structure of these proteins, they are grouped into two topological groups. So one is this beta-barrel OMPs, which are made by these multiple beta strands and embedded into the bilayers of the outer membrane. And the other group is lipoproteins. They are mainly anchored into the inner leaflet of the outer membrane. However, there are also some lipoproteins which can be transported across the outer membrane and anchored on the surface. They are called the surface lipoproteins. Currently, the mechanism of the SLP transportation remains unclear. But on the other hand, the assembling of the beta OMPs is quite well studied, especially in the model organism E. coli. So it has been reported to be mediated through the BAM complex, which is a conserved machinery across the entire bacteria kingdom. So BAM itself is like a 5 protein class. The classical one, BAM, contains 5 proteins. At the center of this complex is a BAM-A protein, which itself is an OMP. It interacts with the 4 periplasmic lipoproteins, BAM-B, C, D, and E, which binds on the portal domains of BAM-A. So together, these 5 proteins insert and fold and insert the substrate into the outer membrane to form these OMPs, beta barrels. So for details how it works, so for BAM-A itself has a lateral seam which is formed by the first and the last beta strands. So once there is a income substrate to be assembled, the lateral gate will open, and the first beta strand will pair with the last beta strand of the substrate. And then the machinery can eventually assemble the remaining beta sheets, and until it forms a complete beta substrate beta barrel and released and inserted into the outer membrane. So there are also some studies indicates the BAM machinery is involved in the flipping of the SLPs across the outer membrane. So in our lab currently, we are working with a bacteria belongs to the Bacteroidota phylum. So this bacteria, they are a large group of gram-negative bacteria. They are evolutionarily distanced from E. coli and exist basically everywhere. So more importantly, so Bacteroidota, they are the major components of our human microbiome. So that's why they are closely related to our health for like a nutrient uptake, and also there are a lot of these pathogens which can cause a lot of disease. So during we actually noticed that there are clear features of Bacteroidota outer membrane proteins, for example, which is very different from other bacteria. For example, lots of these OMPs in Bacteroidota, they are very complex compared to E. coli, for example. They often have very large extracellular domains regions. These are the direct comparison of E. coli OMPs with Bacteroidota OMPs. You can image it might be very challenging for the canonical BAM to handle and assemble these difficult OMPs. So, and also in the outer membrane of Bacteroidota, there are like a very abundant amount of surface lipoproteins. In contrast, E. coli only have less 10 surface lipoproteins. So the question is, standing question is how these SLPs get transported across the outer membrane. So these are the questions we were initially trying to address when we started this work. So using our model organism, model bacteria, Flavobacteria johnsonia, so we managed to isolate and determine the structure of the first BAM machinery from Bacteroidota. From here, you can clearly see the Bacteroidota BAM machinery is very different, distinct from the canonical BAM. It only retains the core components BAM-A and BAM-D, but missing the other three periplasmic lipoproteins. But instead, it has four new, very novel components, two surface lipoproteins, BAM-M and BAM-G, and one beta-barrier protein, BAM-F, and a periplasmic protein, BAM-P, which represents the plug protein, we call it. So in the structure itself, the two surface lipoproteins, BAM-M and BAM-H, they interact with each other, and they are anchored to the BAM machinery through the direct interaction with the BAM-A and BAM-G beta barrels. So together they form a canopy structure, which extends from the back of the BAM-A and all the way towards to the front of the BAM-A, and also covering the lateral seam, which is the activity center of this machinery. So this forms with protective cavity above the outer membrane. And also the BAM-G, the OMP, it's associated at the back of the BAM-A, while in the periplasmic, BAM-P interacts with BAM-D and BAM-A on the portal domains, but also it inserts the middle part of the BAM-P protein in the lateral seam. You can image that when the substrate needs to be assembled, BAM-P must be disassociated from the complex. So when we initially found this very distinct complex, the obvious questions are why the Bacteroidota BAM is so different from the chronic one, and what are the functions of those novel subunits. So to try to address these questions, we try to delete these subunits individually. We found out actually, except this BAM-M and BAM-G periplasmic proteins, all the rest subunits, they are essential. And these subunits, they are also conserved across the entire FCB superphyllum, indicates that they have like a functional importance for the machinery. So let's talk about firstly about the non-essential components. So the first one is this surface lipoprotein BAM-M. So the mutant didn't give us any detectable phenotype, except when we try to purify the protein for structure. So we actually only obtained the broken complex that contains the BAM-A and BAM-DP, but missing the other essential components. So to point out, BAM-G and BAM-H, even they are not present in our purified complex, they are still essential in the background. That means in vivo, they can still form a complex. Indicates the BAM-M is involved in stabilize the entire complex. While in the case of non-essential periplasmic protein BAM-P, so the deletion, again, we didn't see any detectable phenotype, but since it binds at such an important region for the lateral seam, and also some recent studies reported BAM-A specific antibiotics darabectin. It binds tightly on the lateral seam of E. coli BAM-A and blocks the activity of BAM machinery, and thus have a very strong killing for E. coli. However, it's also reported that the Bacteroidota, for example, Flavobacter Johnsenia, which is tested in our hand, they are resistant to darabectin. So we reasoned that it's likely because of the presence of BAM-P, which blocks the binding of darabectin at this region. So we tested this, and it's true. When we actually remove BAM-P and all the BAM-P homologs, Bacteroidota, in this case, Flavobacter Johnsenia, it became to be sensitive to darabectin. And also when we actually overexpress the BAM-P protein in the sensitive background, so they regain the resistant, indicates BAM-P actually is contributed for the resistance of darabectin. So now came with this difficult part about the essential components. Since like BAM-H and BAM-G, they are essential. We cannot delete them directly. So what we instead did is we developed an inducible system, which the essential components are expressed under the control of an inducer. When we actually remove the inducer, the essential protein stop expressing, and the cells will stop growing and lyse in the end, like you can see here. So what we found is when we deplete the essential novel components BAM-G and BAM-H, so we basically observed a very similar phenotype as a BAM-A depletion, which we used as a control because we know this one is essential. And we observed the typical outer membrane assembling defects. Like you can see this is the TM images of cells with and without induction. Without induction, cells becomes the outer membrane became blabbing, and you can also see the leakage of the periplasmic content, while the inner membrane still remain intact, indicates they have very strong outer membrane defects. And also in combination with a lot of other biochemical experiments and also quantitative mass spec analysis of the whole membrane proteomics, we actually figured out that BAM-G and BAM-H, the depletion, they showed a very similar phenotype with BAM-A. As you can see here, a lot of these outer membrane proteins and surface lipoproteins get reduced in the abundance in the outer membrane. So yeah, I mean, this tells us these essential proteins, novel components, are quite important for the function of BAM machinery. But there are two possibilities to explain these results, observations. So scenario one is the BAM-G and BAM-H. They are involved in the core function of BAM machinery. They are required for the folding of all the amps. Once you remove any of them, the entire machinery stops. However, the second scenario is the BAM-H and BAM-G. They are required only for for the folding of a subset of substrate. However, when you remove them, this will lead to the misfolding of these substrates, specific substrates, and this substrate will jam the machinery. In the end, it will also cause the lethal phenotype. So how to verify these possibilities was actually a challenging question that bothered us for a while, for quite some time. But luckily, we managed to answer these questions by selecting some suppressor mutants of BAM-H. What we did is we keep growing the depletion strain of BAM-H in the absence of the inducer, and select the cells which can grow, restore the growth, but without the expression of BAM-H, as you can see here. So we managed to get a suppressor which has a point mutation in the surface loop 6 of BAM-A. So with a substitution of Q801 to lysins, this substitution actually disturbs the hydrogen bond formation between the glycine 509. So we thought it actually disturbed the packing and the structure of the loop on the cap of BAM-A, which might make it easier to release the jammed substrate. So indeed, actually, when we actually combine it with a lot of other biochemical experiments, we figured out that this suppressor mutant, although it allows, it restores the growth and recovered a lot of the synthesis of a lot of outer membrane proteins compared to the depletion, you see. So, however, there is still a group subset of substrate or outer membrane proteins. They are not recovered in the abundance in the outer membrane. Among those, they are mainly these SACD complex proteins, which have like a very complicated surface region, and also TONB dependent proteins with large surface loops, which is supposed to be difficult to handle. So with all this data, it came with some working model for us that the novel canopy of the Bacteroidota BAM, it creates a protective cavity which protects the folding and of these complex OMPs once they are removed. So that will lead to the misfolding of the substrate, subset of substrate, and jam the complex. However, when the loops or the cap or the top of BAM-A has been disturbed, folding packing is disturbed or mutated, like for example here by substitution with the point mutations, although the protein misfolding is still happening, but they probably can release and the substrate get proteinized. But this is not jamming the system anymore, so the cells is able to grow properly. So now come with a question.
Can I ask you to start wrapping up? Sorry, we've gone a little bit over time, but it's a really great story. So.
Just one last slide. So come with this SLP transportation. So we saw some reduction in the SLP abundance in the outer membrane protein, but that could be an indirect effect. To test that, actually we applied proteinistic experiments. In principle, if BAM is responding for the flipping of SLP, so when we remove the SLP, we should expect when we remove BAM machinery, we should expect the SLP to be accumulated in the leaflet of outer membrane. And when we use proteinistic to digest the protein from external, these proteins should be protected from digestion. When we use surface lipoprotein SACE here as a candidate, what we observed that actually is not the case. When we remove BAM machinery, we can still see this protein get transported and chewed up by protein K, indicates BAM is not really involved in the transporting of SLP across the outer membrane. It's likely they have other alternative pathways to transport or transportation. So with this, I would like to thank my supervisor Ben Berks and our collaborators Susan and Justin to solve the structures, and Louis helped me with worked with me for all this biochemical part of this project, and some founders and the facility people support this work as well. And thank you for your listening. I'm happy to take questions.
Thank you so much. It seems like such an enormous amount of work, such an interesting story. It was when you first put the slide up of the E. coli proteins looking so different from the proteins from your system. I had a few questions. I think, to be honest, you've answered them in your talk. So I will move on. Michael, do you want to just ask your question in person?
Sure. And again, anyone else who wants to ask, feel free to raise your hand or put a question in the chat space. You've got priority. But in the meantime, I was going to ask, thinking about Troy's talk as well, do your BAM assemblies operate as singletons surrounded by lipid, or do you think they work as cooperative sets? There's so much going on there. I was wondering if they work as dimers or multimers of these large BAM complexes.
So far, no, from the mechanism of how it works, it doesn't need to form optimal oligomers like multiple BAM machineries itself actually can efficiently assemble the substrate alone. So they have done a lot, actually trapped the intermediates of assembling. So you can actually see there is a BAM-A machinery and with a substrate which is grown out. So I mean, this clearly this machinery is sitting in the outer membrane. It's surrounded by the lipids, like even LPS. So we actually got like two LPS molecules trapped in our substrate, in our complex, but we don't know the function of this LPS yet. It's plausibly so they are helping to maintain the stability or rigidity of this complex because it has been reported in E. coli out-membrane proteins some of these lipids and LPS is stabilizing the formation of protein clusters and patterns. But in our case, we don't know what's the function of this lipids we observed them. Yeah.
Yes. Do you think they're structural, or do you think there's some specificity there that's helping to modulate some movement?
Yeah, that's actually maybe I should. Yes, I think that's one possibility because you see, our BAM machinery has a very huge canopy on the top. So one hypothesis is when they actually try to, it protects the synthesis of the complex substrate. At some point when you need to release, you probably the canopy need to move away or whatever. So I think that's one possibility. Yeah, the lipids either need to push it away or yeah.
Okay. Any other questions from the crowd? If not, I'm also just wondering, in other bacteria like E. coli, are there any canopy proteins that we just missed by not being able to co-purify, or do you think E. coli really doesn't use canopies?
E. coli we don't think E. coli use canopies, but we are certainly missing a lot of the other subunits, which probably in the periplasmic. So E. coli, they don't have this complex substrate. We believe they don't use these canopies, and these proteins they don't exist in E. coli. So it's conserved in the phylum, but not in the protobacteria. So like E. coli or Pseudomonas. So yeah.
Okay. Well, fascinating story. Thank you so much. Any other questions, including about whether or not polymers might be more useful for solubilizing lipid complexes or anything like that? Maybe I will ask that question just because I usually do. In terms of the purification of your system, looks pretty complex. What were the tricks that you needed to purify the intact complex?
So actually, this is we purified from native level, so it's quite stable. We even purified this complex. I mean, when we run the gel filtration, it's run at room temperature. So no, we use the detergent like LMG, so which is gentle enough, but we didn't see much like problems with this purification. So it worked right away. So